All right, thanks. I'll just jump right into it by introducing the focal group. So Saxifragales is one of the earlier diverging, at least named clades in the eudicots. It's about 113 million years old, sister of the rosids, about 2,400 species. For such an ancient radiation, it is correspondingly very diverse, especially in habitat, which was my primary interest in this group. So we have everything from newly added cynomorphs to this group, holoparasites. We have epiphytes. We have aquatic plants, actually quite a few independent origins. Desert, kind of lowland plants. Probably most of the species diversity is in lithophytes, so rock-growing plants of various flavors, especially alpine Arctic, cold, dry lithophytes, or various combinations thereof. This is Saxifraga oppositifolia, probably the record holder for most cold-tolerant angiosperm on Earth. Very low species diversity in the tropics. It does have some. So, I know I called this, my title was something about inverse latitudinal biodiversity gradients. It's a slight oversimplification. On the right here, this is a bunch of Saxifragales communities. The y-axis is latitude, x-axis is species richness. So it's really more bimodal, really nice symmetrical distribution in the northern and southern hemisphere. Maximum diversity about 30, 35 degrees of latitude. Much more diversity in the north hemisphere. Something that will also pop out to you, here's the map of that. Most of the diversity is in montane regions, so it is somewhat driven by topography probably. Mostly just temperate montane regions, so places like equatorial Africa, Indies, very low species richness as well. So in previous work we published earlier this year in PNAS, we documented evidence for a lag between early diversification and later bursts in rates of trait evolution. At the time, we attributed this to basically a consequence of the diversification process itself, where early diversification was mostly niche neutral, filled available niche space, and then there was an impetus to more finely divide niche space in order to be able to diversify further. And so in community ecology, this is called niche packing, basically more finely delimiting that niche space. Very challenging to test with just macroevolutionary methods in a phylogeny. There are methods using spatial approaches to do this, and so that's the topic of this talk. So I had two goals. First of all, to try to spatially map the diversification process using phylodiversity methods, and then to use some methods to try to test for where on the globe there might be evidence for niche packing. Okay, so the data, just really briefly I'm going to go over. It's mostly from our earlier paper. I did update the occurrence data. There's a bunch of new stuff from GBIF. Highly resolved tree, partly based on phylogenomic data, and more than half of species coverage. I think it was somewhere between 60 and 70%. Occurrence records were used to build species distribution models in Maxent. If you have questions about that, after I'd be happy to answer. And part of this was a biodiversity literature extraction project to get morphological traits. So these were vegetative and floral, mostly from floras. So really brief on methods. As far as the diversification, I calculated relative phylogenetic diversity for the purpose of this talk. Really briefly, if you're not aware of this type of research, phylodiversity methods seek to capture how much a community captures evolutionary divergence. So basically it's a measure of diversity that has something to do with a phylogeny. So imagine we have a global species pool and we know all the phylogenetic relationships for a species with branch lengths. And our community members here are highlighted in red. So what we do is we take the subtree of the community members and we calculate some sort of index that represents branchiness or how many small or long branches we have. Relative phylogenetic diversity essentially sums all these branches and then normalizes by the overall tree depth. So we're not capturing something like millions of years. We're trying to get how much sort of shallow divergences versus deep divergences in our community. I also extracted tip-level speciation rates from our earlier paper to try to compare with phylogenetic diversity, because diversification is just one explanation of various types of phylogenetic diversity. Now for traits, maybe this is a little less familiar in this audience. I try to test for niche packing by calculating a measure of functional diversity. Now the traits are not just functional traits, although they include some, so I'm kind of just using this existing literature term. I'm really trying to capture just overall morphospace and quantify that. So I use the convex hull method. Imagine this x and y axis somehow represents morphological variation. In this case, it was Gower's distance, basically. This scatter plot is a community. Each dot is a single species. The convex hull approach finds the n-dimensional polygon, or here two-dimensional in this example, that is of minimal volume, contains all the points, and has no concave angles. And the volume of that structure quantifies how much of overall morphospace is in the community. So this is a test for niche packing because basically you expect these kind of metrics to scale with species richness, and if you have basically for a given hull fewer species than you expect, then that is evidence for niche expansion, and if you have more species than you expect, it's evidence for niche packing. So typically you don't care about the absolute scale of any of these indices. I mean, maybe for some projects you do, but typically you want some sort of a significance threshold, so you know if it's significantly over or under. So I generated 1,000 random data sets, sampling without replacement, and critically maintaining species richness, because as I mentioned, these indices all scale with species richness. It's based on methods implemented in biodiverse and talked about in this paper in Nature Communications, Mishler et al., 2014, but these were all custom implementations. Randomizations use a global species pool, so any Saxifragales species from anywhere on Earth could go into a community. I'm going to come back to that in just a few slides. Okay, so straight to results here. I'm going to use this color scheme throughout the talk. White communities, either for the most part had no Saxifragales species, or they had very small Saxifragales communities so that I couldn't calculate the metric. Gray communities are within null expectations based on that randomization. Blue communities are greater than expectations, so this is showing relative phylogenetic diversity. So these are communities with very deep branches. And red is under expectations, so these are communities with very shallow branches. The first thing I was really excited about is, not always true with a lot of these data sets, but there was really clear spatial patterning to the data. So the first thing that popped out to me is Saxifragales exactly recapitulates pattern documented last year in a letter to Nature on the angiosperm flora phylodiversity of China. So basically they documented evidence that East Asia is a phylogenetic museum essentially with lots of deep branches probably representing tertiary relics, and much shallower branches probably active diversification in Qinghai Tibetan Plateau, Himalayan region. So that's exactly what we see here, so I started to believe my results. Some really interesting patterns over here. We already kind of know that southeastern US has a lot of these same tertiary relics. Probably a lot of these tracks are disjunct between these two, so that made sense. And there's already kind of a sense that Western North America is a sort of younger flora with lots of very recent radiations, and so that's seen here. I don't know a whole lot about African biogeography, so if you guys know anything about this, this sort of East versus Western South Africa and Madagascar being more like Western South Africa, come talk to me if you have any insight on that. Okay, for functional diversity, same color pattern. There's only evidence for communities that are under expectation. So this was evidence for niche packing. So that's good. Probably I'm not seeing evidence for communities with more than expected functional diversity because of that global species pool that I mentioned. So if I can just pick any Saxifragales species, on average, probably that convex hull is going to have most of the morphospace. So one of my next steps is to try to regionalize some of those randomizations so I get some statistical power to hopefully detect some other communities. But I found some evidence for niche packing at least. Okay, coming back to the relative phylogenetic diversity, this is a violin plot showing you, here are our categories under, expected, and over RPD, and the y-axis is diversification rate. So basically, high or low phylogenetic diversity, people often kind of subjectively talk about it as somehow related to the diversification process. I wanted to quantify this, and it does turn out that communities with very shallow branches had high diversification rates, and especially communities that had very deep branches had very low diversification rates. Keep in mind that is a log scale, so those are pretty significant differences. Coming back to the phylogenetic the functional diversity, I wanted to test a couple of predictions that should come with this. So if niche packing is the result of a gradual diversification process, you would expect that older communities should have more niche packing and younger communities should have less. So I quantified this basically in two ways. On the left here, this is on the y-axis suitable climate age. So what I did is I calculated a niche envelope that was basically climatic tolerance for the entire order Saxifragales. I took mean values for all the species and calculated sort of a like a 95% ellipse of climatic values essentially. And then I used paleoclin data that goes back to the Pliocene and did a regression analysis to try to estimate when a particular community enters suitable habitat space for Saxifragales. And so in millions of years, how long has this community been able to support Saxifragales species? So our communities with evidence for niche packing actually are in those older communities. And so that makes sense. And so that's good. The other metric here is temperature Euclidean distance. This is a bit more of a standard concept. I just took all of the paleoclimate scenarios, took the pairwise Euclidean distances, and so this is a metric of basically how much temperature climate change there has been since the Pliocene. And so communities with very high values are very unstable communities. And so it turns out that the most stable communities tend to be niche packed, and so those are probably also communities that have had Saxifragales species for a longer period of time. With that, I would like to acknowledge my previous postdoctoral affiliations at UF, Doug and Pam Soltis and Rob Guralnick, a couple of NSF grants that have supported some of this work, and a shameless plug here, I'm starting a lab at Mississippi State University. If you like big data stuff and plant science and you're looking for graduate or postdoctoral opportunities, come see me. Thanks.